So, hello everyone. It is my pleasure to introduce today's speaker, Dr. Corree Laule. She's a physicist and also an associate professor in the Department of Radiology and Pathology and Laboratory Medicine. She's also a vice chair of the research in radiology and associate director of the International Collaboration on Repair Discoveries, the University of British Columbia. Sorry? No? Okay. And she has been involved with nuclear magnetic resonance and magnetic resonance imaging research for over 26 years. She heads an interdisciplinary MR research program spanning technical innovations to clinical translation, and she's interested in understanding the microstructural and pathological determinants which govern the MR signal changes in central nervous system tissue. And her primary areas of research, which I personally find very, very interesting, are the multiple sclerosis and spinal cord injury. And she has extensive experience in imaging, both in vivo and postmortem brain, and also in the spinal cord, with emphasis on characterizing the myelin. And she is passionate about science communication and leads initiatives to increase the diversity in STEM and emphasizing the women and the indigenous peoples. I would ask you to mute yourselves when you enter. Also, I wanted to mention that the title of her presentation today is the Exploring the Human Brain and Spinal Cord Microstructure with Quantitative MRI and Histology. Welcome, Dr. Laule, and we are very happy to have you with us today.
Wonderful. Thank you very much for that kind introduction. Just to let me share my screen here. That should all be working. We tested it. Is that all good?
Yeah, yes. It looks good.
Okay, perfect. All right, excellent. Get going. There we go. Okay, all right, great. Thanks. So thank you very much for the kind introduction and for the invitation to speak to all of you here today. So I'm going to be giving a bit of an overview about some of the work that we've been doing here in Vancouver at UBC, looking at both human brain and also spinal cord using MRI and also histology. Before I get started with the research part of my talk, though, I did want to do a land acknowledgement. As Norma mentioned, I'm based in British Columbia in what is now known as Vancouver, but this geography and these names that we've given these cities and regions have really kind of only been in use for the last few hundred years or so. And traditionally, if we look at what the territories of BC and other parts of Canada look like based on the indigenous communities which were in these regions for thousands and thousands of years before us, we look something like this. And so I just wanted to acknowledge that I am a settler or an immigrant to this country. I was born in Germany. We moved to Canada when I was small. I grew up in the Okanagan, on the traditional territories of the Okanagan people in the interior of British Columbia. And then the work that I'm going to be presenting to you today was conducted on the traditional unceded territories of the Coast Salish people, which includes the Musqueam, Squamish, and Tsleil-Waututh territories. And so I just encourage people to reflect about these lands and territories and to think about who the traditional keepers are of the lands that everybody who's attending are on. I also wanted to just start with some disclosures. I don't have anything too exciting, no relevant big stakes in pharma companies or anything like that. I do receive funding from NSERC, Craig Nielsen Foundation, as well as ICORD and co-investigator on CHR and MS Society grants. And I am an associate editor for the Journal of Neuroimaging. So I've got some objectives. And so the first is to become familiar with different quantitative MRI methods that are being used to study brain and spinal cord. The second is to learn about postmortem MRI validation studies, focusing on spinal cord injury. And then the third is to review in vivo myelin MRI changes in different neurological conditions. So just a little bit of a background about me before I kind of jump into some of the different research things that we've been working on. So my research program is kind of broken up into 3 different prongs or arms, if you can think of it that way. So I'm, myself and my group and my collaborators and colleagues are involved in MR development. So things like doing coming up with new ways to use the MRI scanner, so sequence development, also analysis development, doing experiments on phantoms, recently doing more NMR work as well, simulations. There's also a validation arm, which is looking at working with human tissue, both brain and spinal cord, where we compare what we measure with MR to different histological staining. And then there's an application arm to my research program, which then uses these techniques we've developed and validated and apply them to different in vivo brain and spinal cord conditions, so including multiple sclerosis and a few others I'm going to touch on. And there's a big, very big army of people that are involved in all of this work. So I've got most of their pictures here, but probably not everybody. So I just wanted to kind of start by recognizing all of those. One of my current PhD students, Alex Emsworth, actually did his master's in your program, and he's a fantastic student. So thank you so much for training him so well. So for today's talk, I'm going to focus on the validation aspect of the work and also the application aspect of the work that I mentioned. But just one quick slide about a primer about brain, spinal cords, and MRI. I realize this might be a bit of a diverse audience, and not everybody necessarily works on neuro or necessarily works with MRI. So just a quick recap, or maybe there's people that are quite new to the field. So if we look at our brain or our spinal cord tissue, and we kind of zoom in at a high resolution, this is an axon, neuron with the surrounding myelin sheath, and then there's an electron micrograph of that. And you can see that there's different structure within this. There's in brain and spinal cord tissue, there's lipids and there's proteins, and then importantly for us in the MRI business, there's lots of water. So there's water in different physical spaces, so water inside the myelin bilayer. And then there's also water outside of the axons in extracellular spaces and many other locations. And so we remember back to maybe some MR basic courses if people have taken those. MRI is primarily measuring signal from water. And in particular, it's the hydrogen atoms that are attached to the oxygen. And this is where we get the majority of our signal, not all of it, but most of it. And so the properties of the water will determine your MRI contrast. And we can look at different things like the amount of water, the movement of water, where it's located, how the water is interacting with non-water hydrogen. And all these different things will give us information, and we can kind of utilize that to get different types of contrast. And so why might we want to use MRI to try and learn about different diseases and injuries and conditions? So we know, using multiple sclerosis as an example, we know from human histological studies in MS, you have myelin loss, you have myelin regrowth, axonal loss. But one of the challenges with these histological studies is that the timescales are they're unknown. Most of this post-mortem human histology work is from people that had very advanced disease, so lived with MS for 40 years. And so we don't really know a lot about the acute changes that are happening. Now, we can use MRI to follow these kind of focal areas of damage, show up often as bright spots or dark spots, depending on the type of image. Sorry, I'll just ask people to mute themselves, please. I'm getting some sound. So, from MRI, you can get these kind of pictures where you follow the changes of these pathologies over time. But one of the challenges is that you see this lesion getting smaller and changing in size and shape, maybe in intensity, but you don't really know what's happening inside. Is there changes in water? Is there changes in tissue, changes in different chemicals? And all these things could be happening in multiple sclerosis. Another example, looking at stroke. So you don't need to be a brain expert or a neuroradiologist to identify the kind of right central area here where the stroke happened. That's the primary lesion site. But Again, the specific type of damage that's happened inside that lesion, you can't tell from conventional imaging. And also, what about the areas that are beyond this primary lesion that look kind of fairly normal based on intensity? Is there damage happening there? And so this has driven us and many other MR research groups around the world to try and come up with more quantitative methods. So because this conventional MRI has this low specificity for focal or bright areas or dark areas that you can pick out, but there's also poor sensitivity for non-lesion disease, so areas that look otherwise normal. And so the thinking is that maybe more advanced and more quantitative techniques might be better suited to try and assess some of these disease processes. There's a whole pile of different techniques that are being explored. I've just listed some of them there. Perhaps people will have heard of some of these before: diffusion, spectroscopy, functional MRI, magnetization transfer, myelin water imaging, and I mean, there's many more as well. And I don't have time to go into depth into all of these, but I will focus on a few of them and tell you about some of the stuff that we've been doing. So for the first part of the talk, I'm going to talk about some of the validation work we've been working on recently. This is a spinal cord injury project, and these are the key people that have been involved with that. So Sarah was a PhD student that really led this work. She graduated recently, and Nikolai has now taken that over. Farah is the histotechnologist on the project, and then Veronica is now taking over as the main neuropathologist for this project. So just a little bit of a background about spinal cord injury. So this is something that affects approximately 90,000 Canadians. There's about 4,500 new cases a year. These aren't necessarily all traumatic injuries, but injuries that affect the spinal cord in some way. 300,000 people in the US and over 20 million people worldwide live with some form of a spinal cord injury. For acute injuries, more than 50% of the injuries are in people age 16 to 30, 80% male, so often doing high-risk sports or other high-risk activities. But interestingly, this is something that I learned when I got involved in this project, and I didn't really kind of appreciate or wasn't intuitive to me, is that the fastest growing demographic of spinal cord injuries is actually people over the age of 65. And this is primarily due to falls. Spinal cord injury has a really high lifetime healthcare cost associated with it. And therapies for acute or chronic injuries provide really, at best, modest improvements. And so there isn't generally a magic pill or a cure that people can take. So here we have a conventional MRI of a spinal cord injury. And you can see the center area of the cord is kind of quite squished there. And then above and below, there's some hyperintensity, so the signal's a little bit brighter. And again, the question is what's happening here? Is there inflammation? Is there edema? Is there myelin damage? Is the axons, are they swelling? Is there axon loss already? I mean, again, we can't tell this sort of information from a clinical MRI. Now, we can, of course, get more specific information from histopathology, but this isn't something that's feasible on people living with disease for obvious reasons, because that would be very invasive, and you want to be taking tissue out of an area that's already injured. And so the question is, can quantitative MRI give us more specific information to help with some of the challenges that clinicians are facing in terms of patient prognosis? If we can come up with quantitative MR measures, they could be used as objective biomarkers for clinical trials. And they might also be able to assess areas that are damaged before treatments are initiated. So perhaps based on someone's tissue damage characteristics, a physician may want to elect to do electrical stimulation versus another type of therapy. And so for this project that I'm going to be describing, we had a number of different aims. We wanted to, investigate a number of different quantitative MRI measures in traumatic human spinal cord injury in terms of looking at validation of histology. also looking at the extent of damage away from the primary injury site. We wanted to see if we could characterize the timeline of damage and also do some correlations with injury severity. For this work, we used tissue from the International Spinal Cord Injury Biobank. So this is a biobank that has been recently established at Vancouver General Hospital, led by Dr. Brian Kwan and being managed by Adam Bellanosi. And so this is a new biobank that just started a couple years pre-pandemic, late 2018, I believe. And so Dr. Kwan's a spinal cord surgeon, and so he's the one who's seeing people as they come into the front lines in the emergency department. And so as part of a bigger research program that he has developed this biobank for people that come up with an injury, and if their injury is too severe that they're either going to pass away or they choose palliative care instead, he approaches them and asks to see if they'd be willing to donate their tissue so that we can do research on it. In terms of some of the MRI techniques that we're using on this tissue, this is just a very kind of high-level explanation of some of the different methods. So one that we're doing is called myelin water imaging. And so this is a technique that looks at the water that's trapped inside the myelin bilayer. This water has a short T2 relaxation time. It's much shorter than water in other spaces. And then from this technique, because this T2 time is so much shorter, we can extract out the fraction of signal from our T2 distribution. So looking at the entire, all the different T2 times, and reflect that as a fraction of myelin water associated signal. And this has been previously validated as a measure of myelin content in multiple sclerosis and also animal models. Another technique that we're using in this study is inhomogeneous magnetization transfer. So magnetization transfer essentially looks at how the water and the non-water are interacting. And this kind of more sophisticated approach of IHMT is proposed to be more specific to myelin lipids, but there really hasn't been a ton of work in this area yet. And then the other method that we're doing is diffusion imaging. So diffusion imaging looks at how water is moving. There's many different analysis and biophysical models out there for diffusion weighted imaging analysis. And so you can extract out information about axons in terms of fractional anisotropy or fiber fraction or axial diffusivity. You can get information that's proposed to be related to myelin, which is the radial diffusivity, how quickly the water will move across the myelin bilayer. There's also models that give information about inflammation and edema as well. So those are the methods that we've been using. In terms of our data collection pipeline, so the whole cord is extracted from the donors with spinal cord injury. This is actually a picture of just a portion of the cord. The injury epicenter is actually at the bottom of the screen, off the screen. So this picture is just part of the normal looking cord. So your spinal cord is actually really small. I didn't appreciate this either until I kind of started doing more in post-mortem work and got to hold one. So it's about the size of your little finger. So pretty miraculous to think that something so small is so crucial in our function. So this is a picture of that. We cut the cord into 4.5 centimeter segments and then do imaging of the methods I just described on our 7 Tesla preclinical system. So this is a magnet that would normally be used for, mice, rats, anything kind of small, but we're using it for our tissue samples. This is some representative pictures and demographic information from the cords in the biobank. So there are 20 to date. So this is just showing 6 of them. As you can see, age is older demographic, as I mentioned. Primarily has been males, primarily has been falls. And I think all but one of the cords in our bank are cervical injuries. So this is the upper portion of the cord. And then there's quite a variation between the time when the person was injured to when they died. Our shortest interval is a couple of days, and then we also have people that have lived for a number of months. We have in vivo clinical MRI, so this is an example of that. And then here's just some representative pictures from our 7 Tesla postmortem MRI. So above the injury, this is kind of typical cross-section of what a spinal cord would look like. The injury epicenter, you can see for some cases, there's really no structure left there at all. It's just kind of this big area of destruction. And then this would be below the injury epicenter. So after we do our imaging, we print 3D custom printed molds for each cord segment. And these molds have knife slots kind of cut into them at 3mm sections. And then our neuropathologist can go in and cut the cord into 3mm pieces. And these 3mm knife slots line up with our MRI imaging. So that's how we can do our matching between MRI and histology. The tissue gets embedded in paraffin and then gets stained for a number of different components, myelin, axons, inflammation, and the slides then get scanned at 40 times magnification. So this is kind of a graphical representation of the histology analysis pipeline. This is something that my student Sarah developed and has now been taken over by Nikolai. But essentially, the histology images are downsampled. Saturation images are created and then thresholded to identify the pixels that are stained. And then the regions are, various regions are manually drawn. And then this gets read out as a stained area fraction and is correlated with the MRI. Sorry, I'm just going to mute myself. I have to cough. There we go. So here's some kind of visual representative pictures of some of the different things that we're seeing with our quantitative MRIs. So these are 3 different techniques that are proposed to be related to myelin in some capacity. So diffusion radial diffusivity and homogeneous magnetization transfer, and then also myelin water imaging. And you can see the contrast between them. There's some similarities, but there's also some differences. And then here's representative images that are proposed to be related to axonal staining. So diffusion fractional anisotropy is a marker that's commonly related, proposed to be related to axons. There's many things that influence fractional anisotropy, and then diffie refraction is another marker that's extracted from a different model that is also proposed to be related to axons. This is just a similar sort of qualitative representation of some of the images, but highlighting 3 different cases. The myelin measures on the left, myelin staining shown in blue, axon measures, and then axon staining, just to highlight some of the heterogeneity that we see between different people. And this is one of the interesting things that we're looking at, and I'll talk about this a little bit later about some of the changes related to injury to death interval and changes that we're seeing away from the primary injury site. So, so moving on into that. So one of the things that we're interested in is looking at Wallerian degeneration. So Wallerian degeneration is something that happens essentially away from the primary injury site. So you can imagine in the spinal cord, there are tracts that travel from the brain downwards, and then there's tracts that travel from the body upwards to the brain. And depending on which direction these tracts are traveling in and where the injury has occurred, you will see what's called Wallerian degeneration, or kind of this dying back of these axons in the tissue after the injury. So if we're looking at tracts that are called ascending, so for example, the dorsal column here, what we see is that above the injury site in the dorsal column, there is a reduction in signal. And if you look at the image below here, you can see it's still quite bright, but above the injury site in these ascending tracts, there's a reduction in signal. If we look at tracts that are descending, so for example, the lateral column, lateral cortical spinal tract, what we see is that the signal is decreased in those areas. And histology is confirming that we do see axonal loss and also inflammation in some of these same areas. So looking at relationships between marker for axons and how those things might change with the time the person was injured to when they subsequently passed away, so injury to death interval. What we see is that people that had a shorter injury to death interval typically have a higher fiber fraction relative to people that lived longer after their injury. So that's on the left side of the screen is the people that passed away quite quickly. The other interesting thing that we see is that there's a more kind of sharper decrease in fiber fraction at the injury epicenter for the people that had the more severe injury. So spinal cord injury has this way of grading how severe the injury is clinically based on motor function. And the most severe injury is called grade A, which is the ones that I've circled here. And you can see for those cases, there's a very sharp decrease at the injury epicenter where the tissue is totally destroyed, essentially. And that correlates to how these people were clinically while they were still alive. We can also look at correlation results. So this is some of our work that is looking at relationships between markers of myelin and myelin staining. What we see is that there's good correlations between different markers for myelin, radial diffusivity, magnetization transfer, and also myelin water fraction, and luxofast blue for myelin staining. So a few key points from this part of the presentation. There's 3 main myelin-related MRI methods that correlate with myelin staining. And so potentially you could use one or all of these in some combination as myelin-related biomarkers in human spinal cord. Wallerian degeneration, so the axons dying back. We can see that primarily in the cords with the longer injury to death interval, so greater than about a month. And this is reflected in both axon and myelin loss. And this could be a way for trying to identify a timeline for detecting downstream axon damage. And then finally, some of our metrics also are related to injury to death interval and injury severity that was assessed when these people were still alive. So ongoing and next steps, we have so much more MRI and histology analysis to do. I've just kind of focused on the myelin and the axons here, and a small portion of that. But we also have inflammation markers that we're looking at. So that's ongoing work. We want to compare our quantitative MRI that's done postmortem with clinical scans. So all of these people had clinical in vivo scans when they first were admitted to the hospital. And so, is there a way that we can relate what we're seeing postmortem with our 70 fancy techniques to changes in signal intensity that are seen on the clinical scans? And then we're also working on a project that hasn't quite started yet, but trying to optimize the scanning for in vivo, and in particular, people with metallic implants, because often after spinal cord injury, people will have some hardware that's implanted, and that can be challenging to deal with when you're trying to collect quantitative data. But these techniques are feasible. They are available at both 1.5 and 3 Tesla. Generally, you can collect decent data above or below any sort of metallic implant location. And in terms of scan time, you can collect cervical cord myelin water data in about 8 minutes, and diffusion can be collected in about 4 minutes. So we currently have a C grant from my research center to assess in vivo reproducibility. So that was an overview of some of the validation work that we've done, and now I'm going to move into applications. And so for the in vivo application section, I'm going to focus on just one particular method, the myelin water imaging method. So just a reminder, this is looking at the water that's trapped inside the myelin bilayers, which has a shorter T2 time. And this has been well validated in human brain and spinal cord tissue for multiple sclerosis, and also different animal models as well. So starting with multiple sclerosis. So this is a disease that affects about 100,000 Canadians, close to 1 million Americans, and over 2.8 million people worldwide. So I couldn't find annual healthcare costs for Canada, but in the US, MS-related healthcare costs are over $85 billion annually. So, a staggering amount. It's the most common non-traumatic cause of disability in young adults, and it tends to affect females approximately 2 to 3 times more often than men. And the reasons for that are still not quite clear. It's a disease where the primary damage is the myelin, but other components of the tissue also get affected, for example, the axons. There's a genetic component to the disease. So if you have identical twins and one of them has MS, the other has approximately a 30% chance of getting MS, which is much higher than a regular sibling where it's only a couple of percent, but it's not 100%. So there's some genes to it, but there's more to it, such as potentially environmental exposures, and they don't really know. So the cause is still unknown, and this is there's huge worldwide effort to try and study this disease from different angles. So if we look at MRIs from somebody with multiple sclerosis, so this is 2 different people, proton density, conventional MRI, and then the corresponding myelin water image. What we see for the first case here, there's these focal lesions that I've circled in pink. And if you look at the corresponding myelin water image, you can see that those lesions are quite dark. You'd say they have little or no myelin remaining. If we look at the second case here, and the corresponding myelin water map, this person also has some lesions that I've circled in yellow. But those same areas on the myelin water image, they're not nearly as dark as the previous case. So while there's some myelin reduction, we would say that there's still some myelin remaining in these lesions. So we see heterogeneity between people, but you can also see heterogeneity within a single person. So here we have some clinical MRI scans, flare and a post-GAD T1, and then the corresponding myelin water image. If we look at one particular lesion here circled in yellow, bright on the flare, dark on the T1, and then on the myelin water image, it's quite dark. So not completely dark, but there's a central dark core. We look at this other lesion that's right beside it. What we see, similar contrast on the flare and the T1, but on the myelin water image, it's still fairly normal in terms of its signal level there. And so perhaps this could reflect differences in lesion age. This was a cross-sectional study, so we weren't able to answer that question for this data set, but that's one potential hypothesis. We can look at kind of try and answer that question about how different lesions might evolve over time by doing a serial study where we scan, the same person multiple times over the course of a number of months. And so here we see, case, I'm going to show two cases, case 1, case 2, not the same cases as in the previous slide. So at month 0, there's a lesion that appears. It shows some enhancement on the T1, which is indicating this is a new kind of lesion. There's blood-brain barrier breakdown. And then over time, the lesion gets smaller. And then here's the corresponding myelin water image. So you can see where that lesion is. And even at month 6, it's still quite dark in its signal. And so there's absence of myelin there. Here we have a second case. It looks similar on the flare of the post-GAD T1. But the corresponding myelin water image looks quite different. So you don't see that sort of dark central core where that lesion is, even though these images on the conventional image look very similar. And these are both, newly diagnosed women with MS in their early 30s. Neither of them are on therapy. And so this is offering additional information that you can't get from the clinical scan. In terms of comparing some of these MR measures with things that might be a little bit easier to collect than an MRI scan. So we've been doing some work looking at correlations between brain myelin water and blood biomarkers. So this is work done by my former master's student, Jackie. And so he looked at lesion myelin water fraction in relation to neurofilament light. And so this is a large cohort study that we did looking at different subgroups of MS. And for this work, what he found was that it was the secondary progressive patients had a correlation with serum neurofilament light, which is a reflection of kind of active myelin axon breakdown in the blood. Very recent work done by another new student, Noah, who's actually a medical undergrad. So he's kind of doing this in his spare time, which isn't a lot of spare time, but he's been doing some great work looking at comparing 24-hydroxycholesterol, which is a marker of neurometabolism in relation to myelin water fraction. And what he's seeing is that higher levels of 24-hydroxycholesterol are related to lower levels of myelin water fraction in the relapsing remitting group, in normal appearing white matter, in lesions, and in diffusely abnormal white matter, which is kind of this hazy stuff, which isn't quite a lesion, but it's not quite normal. So interesting work there, still very much in progress. So also wanted to highlight, this isn't work done by my student, this is my colleague student, student of Dr. Shannon Colin, Shauna. So she compared myelin water fraction with cognition in MS. And what she found is that different patterns of diffuse myelin damage correlated with different impairment in different cognitive domains. So things like processing speed, verbal memory, and also visual-spatial memory. So that was a kind of a summary of some of the MS work, and I just wanted to finish off with touching on a few different diseases beyond MS. So ischemic stroke. This is a stroke that happens when a blood vessel is blocked, and it accounts for the overwhelming majority of strokes, so almost 90% of strokes. And so what my colleagues at UBC have found is that in people with stroke, compared to matched controls, is that beyond the primary injury site, where like there's this big lesion, there's actually diffuse myelin loss that happens throughout the normal appearing white matter. On average, about 14%. And what you can see here in the maps here, even visually, you can see there's like this reduction in the amount of signal that you see on this particular person compared to their matched control. There's also been work at UBC led by Alex Rauscher and colleagues looking at concussion. And what they found is that they scanned varsity hockey players at the beginning of the season, and then followed them over time, and then had them come in for a scan. When a number of them eventually ended up having a concussion, this group of people has a higher than kind of average risk of having a concussion over the course of their season. And so what they found is that at 2 weeks, there was reductions in the amount of myelin in their corpus callosum and also throughout all of their white matter. But even at 2 months, there were some individuals that still showed changes in their brain in terms of some of these quantitative metrics relative to baseline. So this could have important consequences in terms of deciding when players are okay to go back returning to their sport, because I think there's very good evidence now that having these kind of small repetitive brain injuries can have very serious long-term cumulative effects. So I also wanted to highlight this project looking at a correlation between reading ability and myelin. This was a pandemic project. So this was data that was collected like late 2007 and 8, and then just kind of sat there because we all have vaults of data like that we just don't have time to get around to. But this is one of those projects which showed some really interesting results. Looking at so children between the age of 10 and roughly 18, they were scanned with myelin water, and then also had cognitive testing done relating to reading ability, and then they were classified as good readers and poor readers based on their these reading scores. And so this is just a visual representation of kids that were classified as good readers versus poor readers. And you can see that the good readers have much brighter myelin water signal relative to the poor readers. You can see that kind of visually. And then if we do some quantitative correlations between reading ability and myelin, what we found is that there was a number of different brain regions that showed strong quantitative relationships between reading measures such as RAT3 reading, word ID, word attack, and different amounts of myelin levels in regions like the thalamus and the corpus callosum, and also the internal capsule. So the last disease I wanted to just touch on was schizophrenia. So schizophrenia is a psychiatric condition that is actually very common. So it affects approximately 1 in 100 people. And it involves disordered thinking, delusions, and hallucinations. And so unlike multiple sclerosis, where often if you do a brain scan, not always, but often, you can see kind of these focal areas of damage, these bright lesions. If you scan somebody with schizophrenia and somebody who's a matched control, you don't see, kind of anything obvious that would say, okay, this person, I can, this will help me diagnose them, or this person has schizophrenia. Their scans look really quite similar. And so the hypothesis that my colleagues at UBC, Donna Lang and Sean Flynn and colleagues had, was that perhaps there is a lack of functional connectivity in the people with schizophrenia that is being caused by less myelin in their frontal lobes. And so what they found is that on average, people with schizophrenia had a 20% reduction in the amount of myelin in their frontal lobes, and an overall 12% decrease in global white matter content throughout all of their white matter. And again, these are areas that look normal on conventional imaging. What they also found was that in relation to age, in the controls, there was a relationship between frontal myelin water and how old the people were. And this wasn't evident in the people with schizophrenia. And then interestingly, a similar relationship was observed in between frontal myelin water and number of years of education for the controls. which was not evident in the people with schizophrenia. And so, I mean, a lot of the work in quantitative MR and various other imaging metrics is you have, these groups of people where you have, a group of MS and you have a group of controls and you do a group study and you're like, okay, the MS is different than, the controls, or the group is changing over time. But ultimately, we want to be able to use these fancy quantitative techniques on an individual patient level in the clinic. Like that's kind of what we're hopefully aiming for in a lot of our work. And so, but the challenge is that this data, this quantitative data, can be quite noisy. And so, how do you move things to a single subject level? And so, one way that we think that might be possible is through using normative brain atlases. And so, this was work done by my former PhD student Kevin. So he created, a fairly small 50-person healthy subject atlas for brain myelin water. And this is freely available on SourceForge for anybody to download and use as they wish. And so then what you can do with this is that you have this atlas of normal controls. You can then, compare an individual person or patient's data to it and develop Z-score maps to highlight areas that are abnormal. So this is Z-score maps for 3 25-year-old females with multiple sclerosis. EDSS is just how disabled they are. So these are low disability. And on the conventional image, which is the top row, you can see they look fairly similar. But then on the Z-score maps here, the areas of blue are the areas where there's decreases in the amount of myelin relative to the atlas. And so you can see they actually look quite different. And the bottom row shows, greater than, I think it's 4 standard deviations relative to normal. So this is a way that we can actually start to use some of these techniques to get quantitative information about individual patient differences. There's also been a couple of other healthy atlases from people at UBC. So Adam is a student, former student of my colleague Shannon, and then Sarah, before she got involved in spinal cord injury, also created a smaller healthy atlas for pediatrics. And both of these are also available for people to freely use. Kevin also made a similar normative atlas for spinal cord data using 20 healthy subjects. And it's the same sort of idea where you can then compare individual patient data to this atlas to highlight differences, that maybe aren't visible on the conventional scan. And so again, three different people with multiple sclerosis with not too severely disabled, similar disease duration. And these areas of blue are highlighting areas that, where there's reduction in myelin damage, which isn't evident on the conventional scan. And so just to finish off, looking at where this particular technique is going in the next decade, there's been lots of advances trying to collect the data faster. Multi-vendor comparisons are kind of ongoing. This is a little graph that shows, acquisition time. So this technique was actually pioneered at UBC in the mid-90s. And at that point, it took over 25 minutes to collect a single slice of data. And now we can do a whole brain in, 4 minutes using different techniques. Data analysis is also getting faster. Back in the 90s, it would take 2 days to do, a single slice of data analysis, and that's if it didn't crash, which it almost always did. But now you can do, analysis in minutes or less than a minute, and people are using different machine learning methods to try and speed that up as well. But ultimately, we need to be able to have this technique analysis methods incorporated into the MR system tools so that people can just press a button and out pops your myelin water map, like you can do with spectroscopy or diffusion with some of the vendors. think there's broad application for this technique. So we need larger atlases where you can, match people well to age, education, sex, gender, ethnicity, many different things that we need to be considering. It's also important to try and understand the limitations of the technique. So how do things like myelin debris or water exchange impact the measures that we're measuring? Because as with any quantitative method, it's important to understand where things break down and work towards trying to solve those problems. Lots of applications, I think, for potential applications for clinical and therapeutic trials. There have been a couple of multiple sclerosis clinical trials that have used myelin water imaging, and I think there will be more to come. And then, this is a partial list here of all the different diseases that we could be looking at with this technique, and really anything that affects the brain or the spinal cord where myelin might be involved. And so some final take-home messages about myelin water in particular. So this is a method that has good validation as being specific for myelin. There's many different acquisition techniques now, and it's possible on most systems. I think, with the growth and development of this method, having more than one approach to collect the data and also analyze the data is important, because that, will increase confidence in findings, and where there's discrepancies, that'll help us learn about the technique and also the disease populations. There's growing data kind of emerging in development, aging, and disease. And I think, ultimately, with the advent of atlases and multi-center studies, this is I think really a technique that we're going to see a lot of growth in the next decade. There's lots of potential uses in terms of understanding disease processes. Can we watch areas of damage change over time? And, for some people, are they more likely to remyelinate than others? And if so, what can we learn about them? I think it's also important to think about how some of these measures relate to function. So, it's fine to see, oh, this person's myelin is regrowing, but how does that actually relate to how they're doing clinically? Is the myelin functional? And so in doing experiments where we compare myelin water imaging to functional measures such as evoked potentials is important. In terms of monitoring therapy, I think there's lots of applications. Neuroprotection is one, and also there's been a lot of remyelination sort of based therapies that are developing, at least in the MS world. And so with that, I'd like to thank you all for your time. Big thank you to the donors and families for the postmortem validation work that I've described, funding from various agencies. I want to acknowledge my lab and colleagues, of course, and also thank you to Norma for organizing this session and inviting me to speak. It's a pleasure to be here, and I'd be happy to take any questions now, or I'll also pop my email into the chat, and then people can always email me later. So thank you very much.
Thank you very much, Corree, for this super interesting talk, and we are ready for some questions. I will start here with the Glenn side. Do you have any questions for Dr.